Hey everybody, welcome back into another note session with Mr. Lane. I'm Mr. Lane and I'm going to be your guide today. Today we're going to talk about mutations and gene regulation. And some of you are like, oh, mutations? That means like the X-Men and superheroes, Captain America. We're going to talk all about superpowers, right? No. Unfortunately, we're not. Even though I do love the X-Men, you have Colossus there in the center of Colossus. Storm, like seriously, best superpower ever, controlling the weather. Anyway, asking the question, do mutations only happen to superheroes? The answer is quite clearly no. In fact, mutations happen to all living things, and we're going to talk about why that is important. So your learning targets for today is that you can describe, you should be able to say this, I can describe a mutation at the molecular level. So that means at the DNA level. I will define the different types of mutations on chromosomes and genes. Those are the two major types. So what is a mutation and what are the different kinds of mutations? I also want you to be able to say with confidence and gusto that I can explain examples of the effect of a mutation. So first off, you might be asking, what are mutations? What are mutations? And that's a great place to start. So let's do that. A mutation is simply a change in the nucleotide sequence of DNA. That means all you're doing is changing an A, T, C, or G to a different letter. That's all. This can occur in somatic cells. These are body cells, i.e. not your eggs and sperm. Okay, this we have seen already. We know that mutations can cause cancer. When we read in class, we read a story about a gentleman who had lung cancer, and that was caused ultimately by several mutations in several genes. Or these mutations can occur in the gametes. Gamete is just a fancy science word for eggs and sperm. We're going to come back in the next unit and we're going to talk about gametes more than you care to remember. So just stand by for that. So to review, a mutation is simply a change in the nucleotide sequence, the A, T, C, or G sequence. It can happen in body cells, which will affect you as an individual, or it can happen in gametes, which will affect you, your offspring, and generations to come. So you might be asking, well, Mr. Lane, are mutations helpful or harmful? And my answer would be like, well, that depends. They happen regularly. When we talked about DNA replication, we know that polymerase comes through and makes the new DNA, but that polymerase is not perfect. In fact, every time you replicate your DNA, you acquire about 30 to 40 new mutations. That means every time your cells divide, you're building up new mutations in that DNA just from copying errors, So that happens regularly. They also happen just by walking outside. You have sunlight coming in and hitting you. The chemicals in the environment around you are constantly interacting and influencing your DNA. This is why like vaping and smoking is so bad, because that's increasing your rate of mutation. But the cool part is that almost all mutations are neutral. Neutral means they have no effect. And the way I want you to think about this is if you're like a skin cell, like I have a skin cell on my hand, I might have all of these volumes of information to make an entire me, but the skin cell is not using all of those pieces. The skin cell is using the skin cell parts. So if I have a mutation in like how to make a liver in my skin cell, that mutation is never going to affect my skin cell's ability to be a skin cell. So even though we do get new mutations, as long as they are not in parts of the DNA genome that cell particularly needs, it's not going to have a great effect. So as I mentioned earlier, chemicals, UV radiation from the sun, viruses, and copying errors from polymerase all cause mutations. So these are things that are happening all the time. And the best part is that many of these mutations are actually corrected by enzymes. There's another type of polymerase that goes around, it's searching out these mutations and it's checking them and repairing them. So DNA literally checks itself before it wrecks itself, because mutations are bad for your health. So, are mutations helpful or harmful? Well, as I said previously, most mutations are neutral. They have absolutely no effect at all. But if a mutation is going to occur that has an effect, it's most likely going to be a negative effect, a detrimental effect. In fact, more often than not, if you get a mutation, it could cause cancer is going to be an effect. So the X-Men is really a sad, sad group of people because it's effectively a cancer war. They've all mutated and got different forms of cancer. But that's not where we're going to go right now. Some types of skin cancer and leukemia are a result of somatic mutations. So that's why in the previous slide I had the picture of the lobster in the tanning beds. We have all of these people that go to tanning beds for some reason. Effectively, it's a cancer bed. You're like, yes, expose me to extra ultraviolet radiation because I love getting mutations. Stop it. Just enjoy being whatever color you are and leave it that way. Avoid the mutations. So I just said that mutations will most often have a negative effect, but sometimes those mutations actually improve an organism's ability to survive and make babies. What we call that is a beneficial mutation. And if you look here, we have the rock pocket mouse. I'm actually going to put the link for this. You'll see this later, so take a look out for this. But if you think about this, if we have a rock pocket mouse that is this color and it shows up on these dark rocks, that is most definitely not a good mutation. But if the population of light-colored mice, some of them happen to be a little bit darker, that would give them an advantage to live on darker colored rocks or darker colored surfaces. So what that means is that that mutation, that just randomly some are a little bit darker, a little bit lighter, that random mutation gave that mouse a selective advantage in the environment. It allowed that darker mouse to make more babies. And that right there is what we call natural selection. Yo, we're coming back to that in just a very short while. So stand by to stand by. All right, what I need you to do right now is just write a quick sentence on a scratch piece of paper. We're going to use this 2 more times. I want it to be at least 5 words and just push it off to the side. That's what we're going to do. I have my sentence. I will share that with you later. I want you to work with your sentence when the time comes. So I'll give you a moment. Just jot a little sentence down. 5 words at least. Okay, well, this is fun. Here we go. Let's talk about the different types of mutations. There are 2 major classes of mutations. The first type of major class of mutation is a chromosome mutation. This means it affects the entire flippin' chromosome. Now, a chromosome, if we remember, is made up of genes. That there's different genes that make up a chromosome. Okay? So when you change a chromosome, what that means is you are like changing chapters in a book. This is a big thing. All right. The other major type of mutation is what we call a genetic mutation. This means we are changing letters in a sentence. We are changing letters in a sentence. We are not making these dramatic changes like a chromosome mutation. We're simply changing letters in a sentence. They can still have some profound effects, but it's a much smaller change. So we're going to start off with genetic mutations, and then we're going to talk about chromosome mutations. Okay, well, this is supposed to be, that was supposed to be a GIF, but I don't see it working. Okay, well, this was more or less going to be a GIF of walking you through all the different types of mutations, but appears to not be working on my computer. So we're going to go through them all anyway. So a gene mutation is simply just a change, like I said before, in the ATCG sequence. So we're just changing 1 or more letters. It can involve as little as a single nucleotide, or it could be several nucleotides. This is where as again, we have those copying errors, chemicals, viruses, UV radiation, and a whole bunch of other things can cause these gene level mutations. They're interacting with the DNA at the molecular level. There are several different types of mutations. There's a point mutation, which is a single nucleotide. You're only involving 1 letter. There's different types of these point mutations. There's substitutions, and then there's these other class called frame shifts. Those are insertions and deletions. We're going to talk about what that means here in just a moment. A point mutation is that change in a single nucleotide. It includes deletion, insertion, substitution, as I just said, and it's just 1 nucleotide of a gene. So what I want you to look at here is we have the wild type hemoglobin gene right here. And we have on the 5th amino acid, we have CTT. That transcribes into mRNA sequence of GAA, and it translates into the amino acid glutamine. There we go, we can see glutamine. But if we change that middle T in the DNA to an A, that changes the 1 letter in the mRNA to a U. We now have a completely different amino acid, valine. That 1 letter change is enough to change your normal red blood cell to a sickle cell red blood cell. And the reason that is normal hemoglobin, it floats kind of all separate from each other. But that one little change from glutamine to valine causes those hemoglobins to get stuck together. They clump together on one particular point. And if you were in the classroom right now, I'd be breaking out the model and I'd be like, oh my gosh, it's right here. But just know that they stick together and it changes the physical shape of the protein. So one letter difference in DNA causes the entire protein to have a different shape because that one amino acid is different. And the problem with that is that now these sickle cells, they clog your blood vessels. So these sickle cells, because of that one letter in DNA, it clogs your blood vessels and has a ton of other health effects. We're going to come back to this later, and we're going to talk about why sickle cell might actually be an evolutionary advantage. So sickle cell, again, as I've stated several times, is one nucleotide in the hemoglobin gene. A frame shift mutation. This is when we insert or delete one or more nucleotides. This changes the reading frame like a sentence. And this is where I want you to break out your sentence that you just wrote down at the beginning, and you are going to use it. So what this causes when you change the whole frame is the whole protein gets built all jankified. And allow me to demonstrate. So what I want you to do is with your sentence, I want you to point mutate it. First, I want you to try inserting. So I want you to insert a letter at random someplace in the sentence and see if how that changes your message. Try to keep the words kind of the same number of letters or see how it affects your entire sentence. Once you do that, try deleting a letter. How does that change how your sentence reads? Try substituting a letter, changing one letter for another letter. And then, oh, I have substitution on there twice. It's that important. Do it twice. So which of these are frame shifts? As I've already stated, that we have insertions and deletions. They shift the whole frame. Now, here's the moment you may or may not have been waiting for. Here is my sentence. My original sentence was, the fat cat ate the wee rat. But if I insert an A and I keep all the other words at 3, that sentence now becomes, the fat cat had you erat. We've completely changed the message that is being sent, i.e. we've changed the order of amino acids, and therefore that protein is going to have a very different shape. This is how frame shift mutations work. I could just as easily have deleted one letter and it would have shifted everything over one nucleotide. So frame shift mutations, although they only involve one single nucleotide, they shift the reading frame, or i.e. how the ribosome will be creating the amino acid sequence. So here's another example of frame shift, just to show you a real world. We go from ATG to GAA. We have methionine, glutamine, alanine, glycine, and then we take that G out, so this is a deletion, and now everything shifts over, and now we have methionine, lysine, and then histidine. So we actually have completely changed the order of these amino acids. All right, going back now, we're going to go to the chromosome level mutation. Chromosome level mutations are like bad days. So now we are changing the entire structure. We're losing whole parts. We're ripping chapters out of books, or we're flipping chapters around. This is a huge change. So genetic mutations, we were making little point changes, little things, but some of them had big impacts. This one is always going to be like big impact. So the major types that we have here, and these might sound familiar, we have deletions, inversions, duplications, translocations, and non-disjunction. We're going to talk about each of those in that order. First, a deletion. This one sounds pretty straightforward. You're literally deleting a part of a chromosome. So the chromosome breaks, a piece of it gets lost, and then those other ends come back together. So in this diagram, you can see how this original chromosome, this chunk gets cut out, and then the whole chromosome is shorter. This is a bad day, because that's like saying, hey, here's our recipe book. These are the chapters on cookies. We're just going to break those, rip that chapter out, and now here's our new cookbook, and we don't have any cookies. And nobody likes it when you take the cookies. Inversion. This is the chromosome breaks off, flips around, and inserts itself backwards into the chromosome. So again, here we see there's the original chromosome. We see there's our segment, and now it has cut itself out, flipped itself around, and inserted itself backwards. So this would be like if you took a chapter out of a book, flip the chapter upside down and put it back in. Now everything is backwards and upside down. You can't read that. It's not a good day. Some of you are like, but I could read it, Mr. Lang. Well, not if you're DNA. Because DNA it's still going to be an ATCG, it's still going to be a sequence, but everything will be backwards. Everything's not going to be where it's supposed to be. The template strand is now the non-template strand, and it's not good. You can't make your mRNA the same way. So you'd be making backwards cookies. You'd be putting all the preheat in the oven and trying to take something out and then deconstructing it. I don't know. It just would not work. Duplication. This one again, you're duplicating. This is when a sequence is repeated. This actually has sometimes can be helpful for organisms, but a lot of time it's bad. But we do see that duplication is where we see then this one can now mutate on its own. We see that in the evolutionary history of different animals. So this segment comes out, it duplicates itself, and now we have actually a longer chromosome because we have two segments. So now instead of just one chapter on cookies, we got two chapters on cookies. And the cool part is that second chapter, if it gains a new mutation, you could then have this chapter is for making chocolate chip cookies, but that new mutation could say the second chapter is now for making like another kind of cookie, peanut butter cookies. So it could be good. Sometimes it could be bad too. So translocation, this is what I'm going to spend a little more time on. Let's break it down. Trans means apart, and location means from a different area. So this literally means that we're getting parts of a chromosome from two different areas. So we have two different chromosomes that are not homologous. Homologous means that they are the same pair. We have 46 chromosomes or 23 pairs. Think of them like socks. All right, you have pairs of socks. You have a left sock and a right sock. They have the same types of genes on them. They have alleles. We're going to come back and talk more about that later. But if you take one sock and try to translocate part of that sock onto another sock that does not make a pair, that is not a good day. So what that looks like is we have chromosome 4 here, for example, and chromosome 20. Obviously, there's a difference here. But what we do is this part of the chromosome 4 gets cut off, and this part of the chromosome 20 gets cut off, and they flippy-floppy. And now we create this whole new chromosome and this whole new chromosome. Now the genes are in completely different things. This would be like taking an automotive manual and ripping out the part on carburetors and switching it with our beloved cookie chapter from chromosome 20. Now we have an automotive part that has a recipe for making cookies, and we have a recipe book that has how to rebuild carburetors. This is a bad day. Genes are not where they're supposed to be. This is not a good thing. And finally, non-disjunction. This is where chromosomes fail to separate during meiosis. Very similar to metaphase. We're going to come back and talk more about meiosis, so don't get worried about that word right now. But this is when we're making eggs and sperm. If non-disjunction occurs, what this means is that each egg or sperm is going to have too many or too few chromosomes. The most common condition that we see caused from non-disjunction is Down syndrome. Down syndrome is non-disjunction of chromosome number 21. So individuals with Down syndrome actually have 47 chromosomes, not 46 as a phenotypically normal individual would have. And I'll show you right now how that works. This is a kind of a preview for where we're going. Non-disjunction, we have all of our homologous pairs lining up here. And we see that during what we're going to call metaphase 1, that 3 chromosomes go one way and only 1 chromosome goes the other way. What this ultimately leads to is that the eggs and sperm that are produced have 1 too many or 1 too few chromosomes. Now, usually, if this happened on any other chromosome, having too many or too few chromosomes would be a lethal mutation. That would die. That zygote would go through apoptosis. It would say, I do not have the right material. But when you have non-disjunction on chromosome 21, that is still a viable zygote, and that develops into an individual who has Down syndrome. Yeah, it's fascinating. Again, here was a GIF that did not work. So enjoy that blank screen. Here is the same thing that was illustrated on the GIF. It's just an overview. You can pause it right here. It's just showing you what those different parts, the different types of chromosome level mutations look like. There's the centromere. That's what that purple thing is. That's the centromere on each chromosome. So now I want you to take that same sentence from before, the fat cat ate the wee rat, that was mine, and I want you to just practice doing a duplication, do a translocation with another sentence, do an inversion, do another translocation because, double or nothing. Here's what I have. The fat cat ate the wee rat. That was my original one. I did an inversion. And so now it says the fat, oh no, it doesn't even say, it just says the, and then there's like this weird backwards F and this kind of backwards G, backward, I don't know what this is, ate the wee rat. So now that, if this was on a chromosome, that part of the gene, completely janky because it's going backwards the other way. So I don't even know what that says anymore. So you can see it's kind of a big deal. Why else are chromosome mutations cool? Because, it tells us where we came from. If you look at us and all of the other great apes, orangutans and gorillas and bonobos and chimps and us, those four that I just mentioned, they have 48 chromosomes. Humans, we have 46. And some of you are like, well, it's because we're better. But nay. What happened at some point in our fairly recent evolutionary history is our ancestors actually consolidated two of those chromosomes into one. So our chromosome number 2, if you look here at the diagram, our chromosome number 2 is actually made out of two separate chromosomes. In fact, you can still see the centromere of that original chromosome intact. It's no longer used as a centromere, but it's there. So chimps and bonobos and gorillas and orangutans, they have these two pairs of chromosomes, whereas we have snipped them together and now consolidated them into one. It's kind of fascinating because the genes that are on those two separate chromosomes in the other great apes are still found on our chromosome number 2. So even though we have two fewer chromosomes, we still carry all of the information that we have in common with those other great apes. That's just cool. That's just cool. All right, we're going to switch gears here now. So we are done with gene mutations and chromosome mutations. We're going to talk about gene regulations, and this is where I wish I could spend so much more time, because gene regulation is when things get interesting. Genes can be turned on and off. They can also have a volume, so they can get turned up and down. That's going to be important. That really explains a lot of the differences. Like people will say, like, well, chimps or humans are 99% accurate. Well, that's true. We're very similar to each other, but we turn different genes on at different times and at different volumes, and that's what leads to our differences. Not the fact that we necessarily have different DNA all the time, but the fact that we turn those things on and off in a different order. So gene regulation is absolutely important if we are to understand why we look the way we do. So let's take a look at this strand of DNA. We see that there's regulatory sites. We have a promoter site with RNA polymerase. That's kind of a landing pad for the RNA polymerase nose to come down. And then we actually have the gene itself here. So when we say we look at the DNA in something, it's not all making proteins. There's regulatory areas of DNA that are simply the on-off switches. And then there's the information. So let's talk about that. How does this happen in prokaryotes? Dude, this one's pretty easy. Prokaryotes simply coil their DNA when it's turned off. So you can see in the top here that the RNA polymerase comes down, it hits this little loop-de-de-loop, and it says, bro, I can't go any further, and that turns that gene off. But as soon as the right molecule binds to that repressor, that repressor releases the DNA unspools, and, excuse me, the gene is then expressed. Get a frog in my throat. Get so excited about gene regulation. All right. In eukaryotes, that's like you and I, we have something a little bit more. Instead of just having a loop-de-de-loop, we regulate our chromatin, where that chromatin, the histones can unroll or roll tighter on the chromosomes. We also have what we call regulatory proteins. And if you recall, way back to DNA, it had a major and a minor groove. That major groove is where those regulatory proteins could latch right in. And depending on whether those regulatory proteins and what order they're in, whether they're there or not, that turns the gene on or off, or turns that volume up or down. So we have what's called the ta-ta box. The ta-ta box is, as the name suggests, a repeating pattern of Ta. I want you to think of that as simply a regulatory region. It's kind of like a stoplight or a go light. It's a region that's saying, here's where to go. Okay? And that's what helps RNA polymerase find its place. It's a landing pad of sorts. And finally, we're going to talk very briefly about Hox genes. Oh my gosh. Hox genes. These are like the big bosses of the gene area. So this whole time we've been talking about a gene. It gives me my hair color, like my thumb thing. But there are actually genes that tell other genes what to do. So these are kind of the big boss genes. They don't necessarily say, hey, they don't say make a head. They say, hey, head genes, make a head. All right. So these are Hox genes that literally control where the different parts of your body go. So they don't say I make a leg. They say, hey, leg genes, put a leg there. Hey, arm genes, put an arm there. And the wicked cool part about this is that these Hox genes, because they serve such a fundamental purpose, that they are nearly identical across all animals and other organisms, to the point that if you look at a fruit fly and you look at a person, that even though a fruit fly is very different than us, the Hox genes that control this area of the fly are the exact same Hox genes that control the top of our head. So this is not saying that this is the same genes. No, but it's saying, hey, put the antenna here, whereas ours are saying, hey, put the skull there. So these Hox genes are so important to looking at the evolution of different organisms because they literally give us our shape. Example, the Hox genes for snakes. In us, it says stop making vertebra, stop making ribs. All right, you're good. Hox genes say, all right, that's enough. In snakes, they say, bro, you keep making those ribs, you keep making those vertebrae, and then that snake goes, and you get this long, elongated snake that has 150, 200 vertebrae instead of a couple. So that's an example. Another example, which is a little more dramatic, is what would happen if I exchange the Hox genes for antenna with the Hox genes that control feet? Well, what happens then is you get a fruit fly, in this case, not a human, that instead of growing antenna where it's supposed to, it starts growing feet. So what we did there is we simply switched the Hox genes locations. We switched antennas for feet, and the same genes that would normally build the leg in the leg area have now been told build a leg right here. And we see that we have two not very functional legs coming right out of the antenna of this mutant fruit fly. So again, Hox genes, incredible genes that tell you where to put the things that make us us. That is all I have for you right now. I know this was a lot. I'm looking forward to questions. We're going to be using all this stuff again, so I promise you that. This is your moment of monkey. Enjoy. Have a great day.